We are moving into the home stretch, 3 lectures. So the last 3 lectures, we're going to talk about something different. We're today unusual agents, one of my favorite. And then Wednesday and Monday, we are going to break the rule for this course and talk about specific viruses. We usually don't do that because I don't think it's a good way to teach virology, virus by virus. But these last 2 viruses are pretty important. So we're going to talk about HIV on Wednesday and then avian influenza on Monday. Today, though, we're going to address this question: what's the smallest genome size that an infectious agent can have? In fact, could an agent even exist without a genome? And we're going to talk about viroids, satellites, and prions, or prions, depending on how you want to say it, because it's a synthetic name, just like antivirals. Who knows how to pronounce them? We make a separate lecture for these, although we're going to draw upon a lot of the things we've learned in this course to understand how they work. They don't fit into the regular taxonomy for viruses, for sure. Viroids, as you will see, are major plant pathogens. Satellites are viruses that require other viruses for their replication. Whether they're actually viruses or not is being debated. And finally, we're going to talk about prions. What should I call them, prions or prions? Who likes prion? Who likes prion? Prion wins, so prions it will be. All right, viroids first. These are amazing. They're just little pieces of RNA. They don't encode any protein. And they have no capsid or envelope or protective coat at all. They migrate very effectively from host to host. They don't need receptors. They tend to get into cells by mechanical damage or by insects putting them into the cells. And there's a database of subviral RNA. So these are viroids or subviral RNAs, because I guess they're not quite viruses. And there's a database. There's so far 1,742 different ones that have been discovered so far. They are all small, circular, single-stranded RNA molecules. All right, so they're circular, single strands. That's what they all look like, all the viroids that have been discovered. They don't code for anything. If you put them in a plant, they replicate. And there are two families. One family, Pospiviroideae, they replicate in the nucleus of the plant, and the family Avsunviroideae replicate in chloroplasts. Really remarkable infectious agents, just RNA, no coding region. Maybe this is what viruses looked at in an RNA world. So the first one to be discovered is, they have great names: potato spindle tuber viroid, or PSTVD, that's how they're named, viroid VD, discovered in 1967, 359 nucleotides long. And you see here, this is not potato spindle, this is a tomato plant, I believe. These have been inoculated with different strains of a viroid. This one is the control, it's a healthy plant. So they're inoculated when the plants are small and allowed to grow. And this is inoculated with a viroid of mild virulence, intermediate, and very virulent. And you can see the growth of the plant is stunted. And in other viroids, there are various pathological lesions associated with infection. Some of these viroids do nothing, they're benign. And other ones are really important diseases of agricultural plants. So something that's worried about amongst farmers. Some of my favorite ones are here on this slide: the Kadang coconut viroid, which causes a lethal disease of coconut palms. You can see this one here is infected. It's not doing very well. If you like pina coladas, you're not very happy with this particular viroid. Then there's the hop latent viroid, which infects hop plants, which of course are used to make beer, but it doesn't do anything, so you can still make your beer and it tastes the same. So no problem there. And then we have the apple scar skin viroid. Actually, this is what Steve Jobs was thinking of putting on his computers originally, but he decided it was too scary. This is, makes apples look mottled like this one. The apples taste exactly the same as an uninfected apple. But if you see these in the supermarket, I bet you wouldn't buy it because you'd think something was wrong with it, right? So that's why you don't see them there. I often look for these to see if I can find one so I could bring it here and show it to you, but I've not found one. So it just is a viroid with mild symptoms that makes the apples look bad. So once again, they don't encode proteins. They don't even make an mRNA, which makes sense because why would you do that if you don't encode a protein? The RNAs, as you will see in a moment when I show you a picture, they're extensively base paired. So these actually don't look like circles if you look at them by the electron microscope. They look like rod-shaped structures. And some of these viroids contain what are called ribozymes. These are RNA enzymes. The RNA has autocatalytic ability. It can cleave itself, all right? And that's this activity, this ribozyme activity is needed for replication of the viroid. So a nice way to distinguish these from viruses. Viruses are parasites of the host translation machinery. The viruses all make mRNAs. Remember the Baltimore scheme? mRNA in the middle. They need to translate that mRNA by the host translation machinery. Well, viroids don't need to be translated. They don't encode protein. They are instead parasites of the transcription machinery. The enzymes that make mRNAs in the cell are the enzymes that replicate viroid genomes. So kind of a twist on the parasite approach. So here's the structure of potato spindle tuber viroid, which I mentioned earlier, the first one discovered, showing you the rod shape. And this occurs because it's extensively base paired throughout the RNA sequence. And there are various regions on this viroid RNA that have been studied over the years. For example, this region on the left here is important for pathogenesis, for the ability of this viroid to cause disease in the potato plant. So again, there are loops at the end, and most of the viroid RNA is base paired with a few exceptions. How do they replicate? So they enter cells, and they are replicated by host RNA polymerase II, the enzyme in the host that makes mRNAs. And usually concatemers are made. In other words, multiple unit lengths of the genome joined to one another. And these concatemers are then cleaved. They can be either cleaved by a ribozyme, which is contained within the viroid, or they sometimes are cleaved by host nucleases. So ribozymes, again, are very, very famous RNA enzymes discovered in 1981. Tom Cech receiving the Nobel Prize for this discovery. One group of viroids forms what's called a hammerhead ribozyme. This is an RNA secondary structure that looks like a hammerhead, hence the name. And it is able to cleave itself. So the RNA itself is an enzyme. And again, this ribozyme is used to cleave these concatemers that are produced during replication. So one group of viroids encode in a ribozyme. I shouldn't say encode because that insinuates protein. They contain ribozymes in the RNA sequence. And another group uses host nuclear enzymes to cut up the concatemers of RNAs that are produced, again, by Pol II in the host. So amazing. These RNAs get into cells, plant cells. They can be introduced by insects. They can be introduced by farmers rolling over the plants with wheels that are contaminated with viroids from other plants. They can be introduced by planting. You bring in a new flat of plants and they have viroids in them, and that introduces them to the rest of the crop. Here's an example of how a member of the Pospiviroidae replicate. These are viroids that replicate in the nucleus of the cell. And they come in by whatever means. Remember, plant cells have a very impermeable membrane around them, very difficult to get through, and so either a sharp proboscis of an insect or damage gets them into the cell. The RNA, the ribozyme RNA, which is shown here as a circle, is brought into the nucleus where Pol II makes concatemers. So these are 2 unit lengths shown here, 2 copies of the genome. That's a minus strand. And then we think the same Pol II makes plus strands as well. So the incoming genome is simply called plus because it's the genome, but in terms of how I've taught you about positivity, it makes no sense whatsoever because there's no protein encoding here. It's just a way of designating the viroid and the antiviroid. The plus strands are then brought into the nucleus, the nucleolus, where they are cleaved, probably by host cell nucleases, to unit lengths. They get ligated into circles, and then they're exported out. And then they move to the next cell. You know, cells are connected by tunnels, as you can see here. And the components move from cell to cell via these pathways. And eventually, the entire plant will be infected. The avsun viroidae encode or contain hammerhead ribozymes. That's why we're showing it as this hammerhead-like structure. These come into the cell by similar means. They go into the chloroplast. This is where these replicate. We believe that these are also copied, of course, by host cell RNA polymerases, but the cleavage step occurs by the virus ribozyme, maybe assisted by host cell proteins. And then they fold back into a ribozyme, so that's the minus strand. It's copied again to form plus strands, the same concatenamers which are cleaved by the ribozyme, exported from the chloroplast, and then moved to the next cell. So the RNA is recognized by the host cell polymerase, and it replicates and moves throughout the cell. Now, where did they come from? They've probably been around for a long time. They probably entered crops as we learned how to become farmers many years ago, our ancestors, and we started to breed crops, and then eventually viroids were most likely transferred from wild plants that were used to breed those originally. So they're present in the wild most likely, and we have derived all of our crop plants for the most part from wild plants, and that's where they've come from. Now, if you know anything about farming now, we do monoculture farming. We have genetically identical plant lines for whatever it is you want to grow, corn or wheat or any kind of grain. And these tend to have viroids in them. So they're all the same, and so they're all uniformly susceptible to these viroids. And as I've said, they're transmitted. You can clean up your plants and get rid of all the viroids, but if a neighboring farm has viroids in their plants, you can bring it over on equipment, hands, or even from plant to plant, because as plants grow in rows, they touch each other, so there can even be a plant-to-plant spread. How do they make the plants sick? So I showed you some examples at the beginning of plants that were either stunted or miscolored by viroid infection. Well, this is an area of ongoing investigation, but the current idea is that the viroid RNAs are cut up by the machinery in the cell that's used to make small interfering RNAs. So the RNAs get into the plant cell, and the plant cell has an enzyme dicer that chops it up and makes small RNAs, and we think that these then silence host genes, and that's what causes disease in the plant. All right, so it's a viroid-derived small interfering RNA, but it happens to recognize or hybridize to a host mRNA, and that causes degradation of the host mRNA and therefore the pathogenesis. And so that follows that symptom development in plants correlates with production of small RNAs. If you interfere with their production, you can alter the plant so it doesn't make microRNAs, you interfere with pathogenesis. And the pathogenicity modulating parts of the viroid I showed you originally in the potato spindle tuber viroid, there's an area that controls pathogenesis. This is where the majority of these small interfering RNAs come from. So we don't really know why this has evolved to be this way. Has the viroid evolved to produce small RNAs to be of benefit to it, and these happen to cause pathogenesis accidentally? That still is an open question. So these are really remarkable molecules because they are the simplest viruses, I think. I don't see why we can't call them a virus. They're called viroids because they don't code anything. But it's really amazing that they can propagate, they use entirely the host cell machinery, and they can cause disease as well. Not all of them, but some of them can cause disease. So really interesting, minimal infectious agents.
How does the host RNA Pol II recognize RNA? Because I thought it needed a DNA substrate.
It usually does, that's correct. And this is one of the, in very beginning of the course, I told you, remember, RNA genomes can't be recognized by host cell enzymes. I kept saying that over and over again so that you would understand that viruses need to encode their own RNA polymerases, right? what I said. But actually, there are a few exceptions, and the viroids are one of them. And we're going to see another exception also in today's lecture. So viroids and satellites apparently can be recognized by RNA Pol II. So then you may ask, why haven't viruses evolved to be independent of their own RNA polymerases? And we don't know the answer. There must be some other constraint, but it's a really interesting question. So, you know, I can't really introduce that exception at the beginning because then you wouldn't listen. So, but now it's okay because now you're all smart and have learned all this stuff. And it is, which of the following is not a property of viroids? Why didn't you all put capsid, metastable capsid? They don't have capsids, right? They do not encode a metastable capsid. They don't encode any protein. They can't have a capsid, right? They have a ribozyme. They have a host. They're replicated by host RNA polymerase. They have a circular rod-like genome, but they don't have a metastable capsid. Okay. Next set of interesting infectious agents we call satellites. And again, the name is meant to distinguish them from viruses, but this is just a human naming convention. It may not really be relevant, but I guess it's good to help study these. Satellites are small, single-stranded RNA molecules, very much like viroids in the sense that they're RNA, but these encode proteins. Some of them encode structural proteins that encapsulate the genome. Some of them don't encode anything like a viroid, but some of them do, and they're all satellites, so that's an exception there. For the most part, these satellites don't have the genes needed to replicate. All right, so they all don't have replication genes, and some of them don't have encapsulation genes. So in a sense, some of them are viroid-like, but they have enough differences to be classified separately. And the main one is that they replicate in the presence of another virus, which is called a helper virus, which is very distinct from viroids. Yes?
What do you mean by encapsulation genes?
Sorry?
What do you mean by encapsulation genes?
Oh, proteins that encode a capsid,
Okay.
Glycoproteins, or a shell of some sort.
Never mind. Does that mean that some satellites have capsids?
That's right.
Okay.
Some satellites do have capsids, yeah. And others are encapsulated by the helper. So satellites are in general in a capsid, but where the capsid comes from depends on the satellite. So they replicate in the presence of another virus, which we call the helper virus, and the helper provides replication proteins for all of the satellites, and for some it provides the capsid protein. Okay? So satellites are very common in plants also. I don't know why the plants have a lot of these odd infectious agents, viroids and now satellites. And the satellite makes the disease caused by the helper virus different. So that's why I say causes distinct disease symptoms not seen with the helper virus alone. So that the satellite is doing something in addition to the helper to give distinct symptoms. They are typically not related to the helper virus. So they're not simply defective helper viruses that have lost genes and become unable to replicate. They are distinct entities which have evolved to utilize the helper viruses, but there's no homology with the helper typically. Yes?
Are satellites helper virus specific?
Yes, they are helper virus specific, right? So a satellite has its own helper virus, and the helper of one satellite will not help another satellite, correct? So let's look at some of these satellites. This is just a selection to give you an idea. So there are satellite viruses of eukaryotic cells. So for example, this is the way this table is laid out. There's the helper first, and then a slash, and then the satellite virus. So there are some single-stranded DNA viruses called dependovirus. Clever, right? Because they depend on another virus. Or parvoviruses that are defective, and they require adenoviruses or herpes viruses as helpers in order to replicate. These are encapsulated. Here's the genome size, 4,700 nucleotide. The particle is 20 to 24 nanometers, and these are some of the proteins encoded in the helper genome. Then you see a whole series of helper satellite pairs for insects and plants. So we have a B virus and a B virus associated satellite. We have viruses of plants, tobacco necrosis virus, which is quite a famous one and is, as you might guess, agriculturally important. It's associated with its satellite. And again, tobacco necrosis virus on its own will cause plant disease, but with the satellite, the disease is different. So the satellite is modifying the disease. Here's another plant virus, a virus of corn and its satellite and so forth. All right, so they're pairs, as you asked. These are very specific pairs, and one helper doesn't help another satellite virus. And again, get a sense of the genome size. Some of them are rather small, not as small as viroids, because again, they encode a couple of proteins. Some of them just one protein, and some of them a couple. So those are satellites. Now there is one satellite that infects humans. All right, we haven't found any viroids yet. Who knows, we might find one day, but there is one satellite that infects humans. It's called hepatitis delta virus. It's classified as a satellite because it needs a helper in order to replicate, but it has properties of both viroids and satellites, as you will see. Its helper is hepatitis B virus, and it is often found in people who are infected with hepatitis B virus. It's never found by itself. You need to have hepatitis B infection to have hepatitis delta. When it was first discovered, it was thought that it made hepatitis B more severe, but this has turned out to be a controversial finding, and as far as I can see, nobody believes it anymore. So you don't have any evidence that delta is modifying the disease caused by hepatitis B virus. Here's the global distribution of hepatitis delta virus, or hepatitis delta satellite virus. About 18 million people are infected. That's about 5% of the 350 million people who are infected with hepatitis B virus. And it used to be quite prevalent in Europe. The incidence is going down now. The Asia Pacific, some areas having very high prevalence, as you can see here. Again, you acquire infection along with hepatitis B, probably at the same time. You get it from someone who is doubly infected, and delta and B will both replicate in you. And if you happen to get delta by itself, it will not replicate because it requires hepatitis B as a helper virus. This is what the genome looks like, hepatitis delta virus. The 1.7 kilobase RNA, which is extensively base paired, so it looks like a rod, very much like a viroid, right? However, this does encode protein, so it's not technically a viroid, but the structure of the viral RNA looks very much like a viroid. It has a ribozyme activity in it, very much like viroids, and that's shown by this red dot here labeled self-cleavage. So these replicate by making concatemers, as you'll see in a moment, and those concatemers are cleaved by the ribozyme that is in the RNA. So we have the genome, which is packaged into particles, and we have the antigenome. One mRNA is made in infected cells, and this encodes two proteins called delta antigens, and there's a large and a small delta antigen. And these have structural roles. They help to encapsulate the delta RNA genome into the hepatitis B virus particle. So that's a standard polyadenylated RNA that is encoded here in the genome. So this orange part here shows you where the proteins are encoded. So on the right is a schematic of hepatitis B virus. You may remember it's an envelope virus. It has glycoproteins in the envelope. We use those glycoproteins to make a virus-like particle vaccine. Inside is a capsid, an icosahedral capsid, and within that is the gapped, partially double-stranded DNA genome with a protein and a piece of RNA attached to it. This virus encodes a reverse transcriptase. Here is the hepatitis delta virion. It is also enveloped, and it is packaged with the structural proteins of the hepatitis B virus. So the three glycoproteins of hep B, the L, S, and M, are also present in the envelope of delta. So delta can infect cells via the same entry pathway as hepatitis B. But of course, unless the cell has already got hepatitis B in it, the virus is not going to replicate, hepatitis delta that is. Inside is the delta genome, that rod-shaped RNA molecule, and both large and small delta antigen help organize the genome. So small delta antigen is bound along the length of the RNA, and then the large delta is thought to anchor the RNA genome to the envelope. So you see the delta antigens play a structural role. They help encapsulate the delta genome into the particles. So this genome, this delta genome, if you put it in a cell, it will actually replicate on its own, very much like a viroid. As you'll see in a moment, it's replicated by host Pol2. But it will not be packaged unless hepatitis B virus is providing the structural proteins in the form of the glycoproteins. Okay? So that's the helper function provided by hepatitis B. Encapsidation, the replication is provided by the host. So that's why I say it's got features of a viroid, because it's replicated by the host. So here's the viral delta genome. It's considered a minus strand genome RNA. In this case, it is the opposite polarity of messenger RNA, because this does create a message. And then when this gets into the cell, RNA polymerase II recognizes it as a substrate, which is really remarkable because it is a single-stranded RNA. Although it is highly structured, it is double-stranded, and that may be part of why Pol2 recognizes it. Nevertheless, it's copied to make a plus strand, and that's the green molecule here, and concatemers are made, so multiple unit lengths are made, and these are resolved at the ribosome. The red circle is the self-cleaving site, the ribosome site, so you get unit length genomes. Here's the mRNA being made, for example, here. Here's a full-length plus strand being made. So the full-length plus strands circularize, and then they're again copied by Pol2, making minus strands. Full-length minus strands, again, they cleave themselves into unit lengths to form the genome. So this will then be packaged into virus particles. Again, so the replication is all taken care of by RNA polymerase II. It's really remarkable. And just one protein needed for encapsidation. So that's hepatitis delta virus. The last infectious agent with a genome are virophages. And these are relatively newly discovered. I'm not a big fan of the name, but I'm afraid it's here to stay. It is derived from bacteriophage. Phage is Greek for eat. Bacteriophages were given that name originally when they were discovered the early 1900s because they were shown to destroy bacteria. So they were called bacteria eaters, bacteriophages. These are called virophages by the first discoverers who felt that they were, in a way, eating other viruses. But they're not really doing that, as you will see. These are circular double-stranded DNA-containing viruses. They typically have an icosahedral capsid. And these were discovered in cells infected with these giant viruses that we've mentioned only briefly, the Mimi viruses, Pandora viruses, Pithovirus, Sibericum, huge genomes, 1 million base pairs and greater, with huge capsids, over a micron in length, these viruses, visible in the light microscope. So these virophages were discovered in cells infected with those giant viruses. Now the reason they're called virophages is because they interfere with the replication of the helper. That's not always the case with satellites. They don't interfere with the helper, but this class of what I think are really satellites interfere with the helper, hence they got this name virophages. So they don't really eat viruses, but they interfere with their replication. However, the press loves this name and they will not let go of it, so we're stuck with it. So here are some of the virophages that have been discovered and what their hosts are. You can see first at the bottom they are global in their distribution. They're found in both terrestrial and aquatic environments, both freshwater and saltwater environments, all the way down in Antarctica. The first one discovered was called Sputnik, satellite, right? Sputnik. So a little bit of homage to classical virology. This was originally found in an amoeba, and Mimi virus is the helper virus for that. Its genome is quite large, much bigger than satellite genomes, 18,000 base pair, but look, it encodes a lot of proteins. They all have 20 or more open reading frames. Here's another virophage of another giant virus called Cafeteria renbergensis virus, organic lake virophage found down here in a freshwater lake in Antarctica. This is a great one, Lentil virus, found in contact lens fluid of a patient with keratitis. Lentil, of course, the French word for lens. So even the French can name their viruses cleverly. Well, actually, most of these were discovered in France by French scientists. And a bunch in Yellowstone Lake, infecting either Mimi viruses or Ficodna viruses. and then one in another lake in Antarctica. So very large. I was at a meeting last year where one of the scientists working on these gave a nice talk, and he said to me afterwards, don't you think we should call these viruses? Really, it's kind of insulting to call them satellites because they're big and they have a lot of open reading frames. But the fact is they can't replicate on their own. Otherwise, we'd call them viruses. So they are virophages, really a different kind of satellite. And remember, all the satellites I've told you about are RNA genomes. These are DNA, so these are distinct. So they're called virophages. Here's some interesting pictures of cells infected with these satellites along with their helper. So these are cells infected with mimiviruses largely. And mimiviruses are these giant particles, about 800 nanometers in diameter, here, with a fringe around them. They typically have this interesting fringe. And here are the virophages, mimivirus virophage. That's what MVF stands for. You see, they're much smaller particles in there. And this is the factory in which mimiviruses replicate. Right? These are cytoplasmic inclusions where the virus is replicating its DNA and making its protein. And that's where these virophages replicate. They go right in the factory, use the replication systems of the giant virus, and they inhibit their production. And many weird things happen as a result. Not only do they inhibit the yield of mimivirus, but they make the particles messed up. So here, on the upper right, this mimivirus actually contains some of these virophages in them. That's what these smaller particles are, here and here. This one is developing some weird multilamellar structure on one side of the mimivirus particle. Again, as a consequence of having these virophages, here you can see it's wrapped up in that structure. And here it's is a larger version of this structure. So odd things happen in cells co-infected with these viruses. A particularly interesting one that I think sheds some light on the role of virophages in evolution is called Ma virus. This is a virophage of a virus that infects a host called Cafeteria roenbergensis. All right, so the virophage infects a virus, or depends on a virus that infects Cafeteria roenbergensis. Roenbergensis is a marine flagellate. It has the name because it eats everything. So they call it cafeteria, where you can get anything to eat, right? So this is a picture of it up here. So it has a virus that infects it, and in turn that virus is parasitized by the Ma virus virophage. And the study of this virophage has shown that it's probably a gene exchanger. It's probably moving genes among giant viruses. So it will infect a cell infected with one type of giant virus, pick up some genes from that virus and move it to another one. You can tell this by sequencing these genomes. So that's why I call them gene exchangers. Now, there's another interesting one found in Organic Lake. I believe that's in Antarctica. This is a virophage that infects a group of viruses called phycodnaviruses. These infect algae. And the virophages, of course, reduce the cell killing by the phycodnaviruses. All right, and they've done studies and modeling in this lake in Antarctica. And the idea is that these virophages actually can modulate the populations in these lakes. So in these lakes down in Antarctica, there's not a lot of microbial diversity because there aren't many nutrients in these lakes. And in addition, there's not abundant sun all throughout the year, just during certain times. So these virophages have thought to evolve to modulate the populations, the microbial populations in these lakes. So it's attracting many ecologists who like to study these very simple ecosystems where, you know, there's maybe one microbe per milliliter of water and the diversity is very low. And it looks like these virophages are actually enabling the growth of these populations of cells, the algae, during times when sunlight is scarce. So I think these are basically satellites, except they're bigger, they have DNA genomes, they encode more proteins, and they inhibit the production of the helper. But on their own, again, they are not able to replicate. If you have a purified virophage and put it into a cell, you will not get new particles out. You need to have a helper. So in that sense, they're not a bona fide virus. All right, next question is, which of the following statements about satellites is correct? Number 4 seems to be ahead, but we got a smattering of other things. So let's see what confusion is. There are no human satellites. That's not correct. Delta is a satellite, right? They are defective viruses derived from the helper. No. I had a slide which said they are not defective viruses derived from the helper. They are unique entities that have evolved separately. They have evolved to be dependent on the helper, but they're not related to it. Like viroids, they do not encode protein. Satellites do encode some protein, as I've shown you. In plants, they cause symptoms distinct from the helper. That's the correct answer. So the helper causes its own set of symptoms. When you have a satellite present, they're different. Okay. All right, the last thing I want to talk about are infectious proteins. What did I say we're going to call it? Prions? Prions? Prions. Yeah, you elected prions. That's right. Now here's Prion Road. I really like that. This is actually a real road sign in the UK, no through road, because as you'll see, if you have a prion, I can't even remember, prion. If you have a prion disease, it's the end of the line, because there's nothing we can do for you. Prions are really, is that wrong? Prions? Prions?
Class like the Prion.
Prion? Why can't I remember
You're
it?
the teacher. So.
No, I want to do what you guys like. Prions. Oh, which means you don't want a final, right? Prions. Mad cow disease, BSE, Creutzfeldt-Jakob, Scrapie, Kuru. These are all things that are pushed around in the press, and we'll talk about what they are. And the Nobel Prize in Medicine in 1997 went to Stanley Prusiner for discovering these and for really pushing the fact that they are infectious proteins. You may be asking yourself, why are we having this in a virology course? Well, you know, when Prusiner first discovered these and started to talk about them, nobody would listen except the virologists. So I think it's only fitting. In fact, virology textbooks still talk about prions. So that's why it's here. And plus, it's amazing. It's just really interesting. So prions or prions cause transmissible spongiform encephalopathies, which is a really long name for a disease of the brain, encephalopathy. They're transmissible, so it's infectious, got an infectious component. And spongiform refers to the pattern of pathology in the brain. We'll see a slide of that in a bit later. So they are fatal neurodegenerative disorders, uniformly fatal. If you have a prion disease, you will die eventually. And we have thousands diagnosed in people globally every year. 1% of which arise by infection, so these can be transmitted, as the name would suggest. But there are other ways to acquire a TSE, a transmissible spongiform encephalopathy, as we'll see. In 2002, for example, 120 people had contracted one of these TSEs, which is called Creutzfeldt-Jakob disease, by eating meat. This happened largely in the UK, by eating meat from animals that had themselves a TSE called BSE, or bovine spongiform encephalopathy. So the people who ate the meat didn't know it came from cows with TSEs. In fact, the people who sold the cows didn't even know they had TSEs because they didn't have any symptoms. The incubation period is so long that you don't actually know it, that a cow is sick until after you've eaten it. So there was an outbreak of mad cow disease, is what this was in 2002. We'll talk about that. TSEs affect all animals, as far as we can tell, not just humans. Cows, in cows it's called BSE, or mad cow disease. In deer or elk or moose, it's called chronic wasting disease. In Nyala, this is a Nyala, I didn't know what a Nyala was, so I found a picture for you. In other kudu, these are called exotic ungulates. They can get their own disease. Cats can get it. Sheep and goats can get it, mink can get it. And in people, we recognize a number of different variants which have different incubation periods and different symptoms and different pathologies. Creutzfeldt-Jakob, fatal familial insomnia, Gerstmann-Straussler syndrome, Kuru, and variant CJD, which was the disease most likely caused by eating cows with mad cow disease. Now, spongiform, transmissible means infectious, and we'll talk about how it's transmitted in a moment. Spongiform refers to the fact that the brain of someone with a TSE has a sponge-like appearance. It has holes in it. And associated with this is psychomotor dysfunction. So you have increasing difficulty not only walking, but processing information. And again, those different names that I just told you, they have various symptoms depending on what part of the brain is damaged. But each disease is characteristic. Creutzfeldt-Jakob was different from Kuru, et cetera, et cetera. So that's a section of brain from someone with a TSE, an advanced TSE, obviously after death. And you can see this is completely abnormal. Not only are there long fibrils of protein, which are the prion protein aggregating, but there are also holes as well. And that's where the name spongiform came from. The first TSE to be recognized was called scrapie. And this was found by farmers in the UK who noticed that their sheep would rub themselves on fences and scrape off a lot of their fur or hair, whatever it is that sheep have. And they gave the name scrapie. And these animals have motor disturbances, much like human TSEs, trembling. In fact, in Canada, it was called tremblement du mouton. That was the name given to it initially because the sheep are trembling uncontrollably. They get paralyzed, they lose weight, and then they die. 4 to 6 week, very quick in sheep, but much longer in people. So this has been known for over 250 years in Europe as a disease, but what the etiology was wasn't known. And in some countries, it continues unabated. In the UK, 1% of all the sheep in the UK develop scrapie every year. And there's really nothing you can do about this. We have no way of stopping the disease once it starts. We have problems even diagnosing it. We don't have a good diagnostic yet because it's just a protein after all. We can't do PCR, although people are developing some methods. And by the time you see it, it's too late to do anything with the animals. So here's a sheep with scrapie, and the only thing you can see here is that it seems very unhappy, and it's lost a lot of its hair, fur or hair by rubbing itself on fences. So again, this is a 100% fatal disease in sheep, probably transmitted from animal to animal, or perhaps even out in the fields through contaminated fecal material in the pastures and so forth. Now the farmers actually started to figure out what was going on years ago. They found that if you introduced a sick animal into a healthy herd, it would transmit scrapie to the rest of the herd. So they thought very early on that this must be some kind of infectious agent. In 1939, they did the virus experiment. They took brains from sheep who had died of scrapies, homogenized them, and put them through a 0.2 micron filter, which would retain everything but viruses. And they found that you could take the filtrate and inoculate it into new sheep, and they would get the disease. So it passes through a filter. So there was some thought that maybe this was a virus infection. However, very quickly after these initial studies, it became clear that the agent, and remember in 1939 we know about viruses already, the agent was really resistant to things that would damage nucleic acid, like ultraviolet light, ionizing radiation, formaldehyde, so resistant. So doses of these that would inactivate nucleic acids had no effect on the agent, whatever it was that was passing this disease. So the idea began to emerge that there's no nucleic acid in this agent. No nucleic acid. This was sort of heretical, right? Because the viruses and bacteria have nucleic acids. And but the fact that this passed through a filter was thought to indicate that this, in fact, was a virus. But this is where Stan Prusener and others began to champion the idea of no nucleic acid, and they weren't really accepted. They were thought to be off base, and even to this day, there are critics who don't believe this is true. But I think the evidence is overwhelming that these are infectious proteins. So this is an experiment that was done that shows the resistance of these agents to ionizing radiation. So what this is a correlation between the molecular weight of the nucleic acid, and we're looking at different viruses here, DNA viruses and RNA viruses on the two curves, and increasing doses of irradiation and RADs here. Okay, so this is just irradiation on the bottom. And then we're looking at the infectivity of the genome. So basically, this is the infectivity, this is the amount of irradiation need to inactivate herpes simplex. So herpes has a big genome, so it's a big target. You don't need a lot of irradiation to inactivate infectivity. When you irradiate the genome, it causes breaks or crosslinks, and the virus is not infectious. As the virus gets smaller, you need more and more irradiation to inactivate the infectivity. So here's SV40, remember, a small circular DNA containing virus. Small target, so you need a lot of radiation to inactivate its infectivity. Same with RNA viruses. The bigger ones need less irradiation than the smaller ones. But here is Scrapie. Scrapie fits onto both curves, but it's way up here at the end. It needs a lot of irradiation to irradiate its biological activity. And this, of course, is an assay where you irradiate and then you put it into an animal like a sheep and you wait for disease to develop. So one of the experiments that showed that there probably is a nucleic acid in these, because if there is, it's more resistant than any other viral nucleic acid that we know about. So human and animal TSEs are very similar histochemically. You have a number of appearances that overlap between sheep and cow and human TSEs. You can see some of them here. Vacuolization of various cell types in the brain. Neuronal loss, which accounts for paralysis and psychomotor dysfunction. The spongiform appearance is the same in animals and in people. The accumulation of protein in clumps, and amyloidosis is another characteristic of these diseases. Fibrils of amyloid protein form in the brain. So again, human and animal TSE is very similar. The way we eventually detected the agent was by injecting organ homogenates into species, as I've told you already, with sheep. And then after months or years of incubation, you see the symptoms, the typical symptoms of the TSEs. Now in sheep, it's a relatively short incubation period, but they're not a very convenient animal model. And so people have moved into rodents, hamsters, and so forth, in which the incubation period can be over a year, depending on how you do the experiment. So these are really, really arduous experiments. When you inject organ homogenates into these animals, they accumulate in the lymph system, and then they spread eventually to the CNS. So the protein makes its way into the CNS. And then you get the pathology in the CNS. Now, interestingly, unlike any other infectious agent that we've talked about, there's no immune response whatsoever. There's no inflammation. There's no innate response. There's no antibody produced. There are no T cells, no natural killer cells, macrophages. And that's because this is a normal cellular protein. Why would you get an immune response against it? And you'll see how that works in a moment. So the problem with TSEs today, and I said they still occur in the numbers of thousands globally, is that you don't know someone has a TSE until they develop symptoms. And then you see psychomotor dysfunction, and you can get a diagnosis of a TSE in a variety of ways, but then there's no way to treat it. We have no treatments, we have no drugs, we have no therapies. So in people, it's, as it is in animals, it's invariably fatal. So this is obviously an area of active research. Although the number of cases are small, still you would like to be able to sort out ways to treat them. Way back in 1967, before Stan Prusiner, Griffiths said, I think these are proteins. These are infectious proteins, and that's where the name came from. I think actually Prusiner developed the name prion. Proteinaceous infectious particles. And he identified these complexes in the brain of Scrapie sheep, and he purified the protein to homogeneity and transmitted it to other animals to prove that it was a single protein that was causing the infection. So prion. It's encoded by a gene that's in all of us called the PRNP gene. And if you don't have this gene, you don't get TSEs, but unfortunately in humans, as far as we know, there isn't anyone that lacks this gene. You can knock it out in mice, and the mice seem to be okay, and they won't get TSEs if you inject them with a mouse prion protein, for example. Now back here in the 80s and 90s, people still said, well, prisoner, your protein is contaminated with nucleic acid and you just can't detect it. So he had to work very hard for many years to silence those skeptics. And then with the advent of cloning, he was able to show that you could take the gene, the PRNP gene, make the protein, and then administer it to an animal, and it would cause the prion disease. So, but still, people say, there's some nucleic acid in there from the clone, and that's what's causing it. So it's actually reminiscent of the old debate over what the genetic material was. So you remember that people thought initially in the 30s and 40s that genetic material was protein, because there are so many different amino acids. It couldn't be nucleic acid because there are only 4 different bases. And for years after the transformation experiment of Avery, McLeod, and McCarty, people said, your nucleic acid's contaminated with protein. And you get the same, now it's the reverse, but they told Prusiner for years, your protein is contaminated with nucleic acid. So the current view is that the pathogenic protein is a conformational isoform of what our normal host protein is. So PRP, prion protein cellular, is the normal protein that has a function in us, in our CNS. It's normally folded, but when it gets misfolded, it becomes a conformational isoform. It becomes pathogenic. So the normal protein is on the outer surface of neurons. It's linked by a GPI anchor, and it has a particular role in neurons. But under some conditions, it misfolds, and then the misfolding propagates. All right. So you can introduce an abnormal conformer into an organism. So you take a sheep with scrapie, purify the protein. It's abnormally folded. You inject that into other sheep. It causes the normal protein in those sheep to misfold, and it propagates. You get more and more proteins misfolded until the disease develops. All right. So the abnormal conformer, which we call PRPSC, in honor of the scrapie origin of our understanding, this catalyzes the conversion of normal cellular PRPC into the pathogenic conformation, the conformationally altered isoform. This is really a remarkable way that it works. So we have a normal protein in us. If you acquire the abnormally folded protein, it will cause all of your normal protein to misfold, and then you develop the disease. All right, so that's illustrated here. Here is on the right the normal structure of the PRP, prion protein. The pathogenic conversion involves the conformational alteration. Lots of beta strands here you can see, and that's the pathogenic conformation. And if you mix this PRPSC with PRPC, even you could do this in vitro, it will cause PRPC to misfold like PRPSC. You just need the protein present. You don't have to have it co-translational or anything. Just mixing the 2 proteins will cause this conformational change. So this is also reflected in the biochemical properties of the protein. Here is the PRPC protein, the normal cellular protein here, with just some motifs indicated here. Here's the GPI anchor that links it to the cell membrane. If you digest it with a protease called proteinase K, it gets completely digested into small bits, small amino acids. The conformational altered protein, PRPSC, when you treat that with proteinase K, it's relatively resistant to digestion. Only the N-terminus is digested away. So this was early on a diagnostic for seeing if an animal had scrapie. You could just take a brain extract and digest and do a Western blot, and you could see that if you had this proteinase K resistant form, it indicated that you had PRPSC present. So that's what is happening during development of the disease. The PRPC is going to PRPSC. So I've already told you that one way you can do this is by introducing PRPSC into the organism. Yes?
Is there a mutation associated with the misfolding, or?
Sometimes. I'm going to get to that. Yes. Yes.
So why doesn't the body see the PRPSC as altered cells?
Right. So the question is, why don't we see PRPSC as altered? And I don't know the answer to that. It sees it as self, so it doesn't make an immune response. I guess that during thymus development, there must be some of this present, and so so the T cells that recognize it are eliminated, and so it's recognized as self. But it's an interesting question. Don't know the answer. Yeah.
Would it be impossible to make a vaccine because the thing that was probably polarizing
Right.
would be pathogenic?
Yes. So could you make a vaccine? So I think the answer is no, because this is recognized as self, although you might be able to couple it to something to make it unique enough to generate an antibody response which would be specific to this conformation. People are trying to do that. Yeah.
How
Yep.
does, you said the PRPC somehow changes the PRSC changes regular proteins to like, from PPRC to like PRSC. How does that happen?
So how does PRPSC change this? This is the million-dollar question that people are trying to answer. It must interact with it, but how it converts it is absolutely unknown. The protein obviously has the ability to have multiple conformations, and how it's transmitted isn't known. But as you'll see later, this is actually not a unique phenomenon. There are other proteins that can change their conformational states.
And that protein, one more question, that PRPC, is it possible that in the cell it can also have like different, is it ever found regularly in the cell as PRPSC?
Okay, so that's a good question. Do you ever find PRPC and PRPSC in the normal cell? It hasn't, but I'll bet it's there in very low quantities, and that's part of the answer as to why you probably can't make antibodies against this, because it's recognized as self. I suspect there's a dynamic equilibrium of PRPC and SC in us, and that's probably important, but for some reason, at some time, the conversion goes one way and you get disease. So if you make mice that lack both copies of the PR and P gene, these are resistant to infection with mouse prions. So you can introduce prions into animals. We've talked a lot about that. You can take scrapie prions from the brain of scrapie mice, scrapie sheep, and inject them into other sheep or even mice, and you will get development of the disease. But also rare mutations in the gene which occur can lead to conversion to PRPSC. So whether you infect with the protein or you have these mutations, the protein accumulates in the brain, and that's what leads to the symptoms. So there are three types of spongiform encephalopathies. Then there's the infectious or transmissible kind, which we've talked about largely. It goes from animal to animal. You consume infected meat, you get a TSE. There's also familial spongiform encephalopathy. This is caused by an inherited mutation. So one of your parents has a mutation in the PR and P gene. You inherited it and you develop a TSE. But maybe your parent didn't, so the contributing factor is not really clear. Then there's sporadic encephalopathy, where there's no mutation, you haven't eaten any contaminated material, it just develops. The protein must misfold incorrectly, and then once there's enough of the misfolded protein, it just keeps going on. So we think that you can find all three of these types of TSEs in animals, and you can transmit it by the diseased tissue. So no matter how the TSE emerges, by infection, familial, or sporadic, you can transmit it because the PRPSC protein is infectious. All right, and what we think is that throughout animal development, these have arisen spontaneously, and then animals consume other animals and transmit the infection. And so probably we acquired it originally by a mutation, and we also acquire it by eating infected animals. Yes?
For those people who got the disease from eating infected meat,
Right.
does that mean they weren't cooking it properly, or does heat?
Oh, you could cook this for 2 days and you wouldn't. And this protein is so resistant to autoclaving. That's one of the it was originally transmitted by corneal transplants, because even if you autoclave the tools, you wouldn't destroy the infectivity of this protein. It's highly resistant. Yes?
Could it be also like a problem with the folding chaperones that help the proteins fold up?
So could the chaperones that are needed for folding be partly responsible for the misfolding? I think that's an idea, but I have never seen anything that points to a particular chaperone that's involved. So I'm not sure if it's correct or not, but it's a good idea. Yes?
So in the labs, people who are working with PR proteins, how do they destroy it? I mean, because it's so dangerous.
I try and make it in a lab. They don't want to destroy it.
Yeah, I mean, whoever's like working, figuring out the structure
Yeah.
and everything, how do they handle the thing?
Well, you have to be careful. I mean, the way that most people acquire these is by ingesting, eating cow meat, for example, or getting a human product like a blood transfusion, a corneal transfusion. Human growth hormone made from people was contaminated with prions years ago, and that transmitted a lot of the disease. Very difficult to acquire in the laboratory. If you do standard BSL-2 practice, it's fine. You can boil it in 9 molar acid and destroy its infectivity if you want to. So there are ways to do it, but you know, these are not normal ways of getting rid of it. So some human TSEs, kuru, iatrogenic, spread by transplantation, as I've just said, infected corneas, hormones, transfusion from patients with, before we knew about this, we were giving blood from people with TSEs before their symptoms, and people who got the blood would get it. Feeding infected animals to cattle, we'll talk about that in a moment, and that's called variant CJD. So the first human TSE was kuru, which was the fatal disease found in the 4 people of New Guinea, right here. This is at a 30-year incubation period, and a virologist from the US, Carlton Gajdusek, went there to study it, and he figured out that it was spread among women and children through them eating the brains of their relatives who died. So this was a ritual in this tribe to eat, the women and kids only would eat the brain of the people who died, and they transmitted kuru. So he convinced them not to do this, and so now there's no more kuru in New Guinea. But that was the first one that he sorted out. In cows, forced cannibalism has resulted in bovine spongiform encephalopathy. The cows, as you will see, so there was an outbreak of BSE in British cows. Here's a cow with near-end TSE. It's because these cows were fed meat from animals that also had TSEs. Cows for many years were fed processed animal byproducts. They would basically blast all the flesh off of a dead sheep or a pig or something else and grind it up. It would be a protein supplement to the cow, so it would grow faster, because if you just let cows eat grass or corn, they don't grow so fast. You give them some protein, they grow really quickly. In the 1970s, this is called meat and bone meal. The processing changed so that scrapie proteins could now survive and pass into cows. So previously there was a process that involved ways that would inactivate the protein. So there was an outbreak of BSE in cows, and this clearly came from having, for example, sheep meat with scrapie in it in the ground up meat that you were feeding to the cows. However, a lot of this cow meat got into the human food chain, and that resulted in variant Creutzfeldt-Jakob. So again, the problem is that you don't know that a cow has TSE when you slaughter it to bring to market. So here's an epidemic curve of this outbreak. About 1.2 million cattle were estimated to be infected. So here's the problem. The incubation time is 5 years for TSE before you see a cow looking like that cow in the previous picture. But you slaughter them between 2 and 3 years after birth. So you know that there are prions in those animals. So here's the outbreak. These are the number of cases in the thousands starting in 1987. And immediately after several thousand cases, it was realized that it was coming from the meat and bone meal. It was banned, but it was too late because there's such a long incubation period. So the epidemic in cows, BSE, peaked and went down, but then the outbreak started up in people. Again, a long incubation period, and then that peaked and went down. So why is it so long? Well, I think it takes a long time for that pathogenic protein to convert all the endogenous protein into a state that gives you diseased tissue. Unfortunately, we still have BSE in cattle. I remember teaching this course in 2012, and the day after this lecture, in the news, there was a cow in California had been discovered with BSE.
How?
How? How was it discovered? It had signs of TSE, so I think this was an older cow that was allowed to get old enough so that it would show symptoms of disease. In 2003, there were 1,390 cows found, and I think lots of cows probably have it, and we're just slaughtering them before we see it. We think that the cow disease is sporadic, so in other words, the protein misfolds in the cow and they get the TSE, so it's not genetic or transmitted. So we need to protect the food supply, but the problem is we don't test most of the meat. Less than 2% of the meat that we make here and in Canada is actually tested for the presence of prions. Even though we have diagnostic tests, it's very expensive to do this. So in some countries, every animal is tested for BSE, but not here. And people are looking for drugs that would block the accumulation of these prions, and that may be something that works out in the future. Sporadic Creutzfeldt-Jakob, somewhere less than 1 million people worldwide. It typically affects older people. This is about 65% of human TSEs, and as I said, this happens with no warning. Abnormal genes, and of course you can transmit it to others if you have a developing sporadic TSE, but you don't have symptoms and you donate a body part or you donate blood or something, you could transmit it. And that's how we think Kuru was established in New Guinea. So that first person who had a sporadic Kuru in New Guinea, the women and children ate that brain, and that started to spread it in the population. Sorry. Familial is an inherited disease. This is associated with autosomal dominant mutations. We have a whole list of specific amino acids that predispose this. And this, again, can be transmitted to other people if you give human products to them. So again, to summarize all of this, we have a normal protein, PRPC. If you eat meat that is contaminated with a pathogenic prion, that's what this means plus here. You convert this PRPC into PRPSC and you develop the disease. That's the infectious form because you're eating a protein which is causing the disease. The genetic form or the familial form, you have inherited a mutation from your parents that causes misfolding of the protein. And then there's the spontaneous disease. The protein spontaneously misfolds. Maybe it's involving a chaperone. Maybe there are other factors. That's the sporadic. All three, same disease outcome. All three can be transmitted if material is transmitted to another animal. Now, there is a species barrier that is been recognized that if you put diseased brain material into the same species, like mouse to mouse or sheep to sheep, you get disease, but different species inoculation is inefficient. So, for example, if you go from sheep to mouse, it takes a long time to get the disease. So the sequence of the protein in the donor and the host should be the same to get maximal efficiency, and we've recognized this species barrier. So if you put cow PRPSC into mice, the disease is inefficiently generated. But if you make transgenic mice that synthesize bovine PRPC, and then you give those animals BSC prions, they now get efficiently infected. So having the same protein as the donor is very important for efficient infection. We don't quite understand that, but probably has something to do with how the protein misfolding is catalyzed. The problem is that cow prion has a broad host range. It infects many other meat-eating mammals, including us. That's why we got BSC from cows. So the species barrier here is not great. So some prions can overcome the barrier. And that's why we worry about BSC, because cow BSC can readily go into people. The disease in deer, elk, and moose is on the rise. These are areas of Canada and the US in yellow where there are infected cervid populations. These are cervids. And if you go to this website, it tells you if you are a hunter what to do. If you hunt a wild deer. So this deer could have chronic wasting disease, which is the name of this TSC. You wouldn't know it because maybe the symptoms haven't developed yet. So if you take the animal apart in the field, they say stay away from the spinal cord and brain. Stay away from the lymph nodes where the pathogenic proteins are likely to be. So the horror is that it's going to get into into humans via consuming deer, because as you know, people do hunt deer in these areas, and even some infected deer in New York State. 90% of standing herds of deer and elk are positive for TSEs, and wild animals as high as 15%. We don't know how they're transmitted. Maybe it's excreted in the feces and the animals eat it, or maybe saliva contamination. But in the lab anyway, you can transmit the deer disease to cows. So people are worried that cows are out to pasture, and at night the deer come and they eat the grass, and they may contaminate the grass with saliva or feces, and then the next morning the cows come in and they pick up deer prions, and develop TSE, and then eventually gets into the food chain. So there's some concern about this, as you might understand. So this just gives you a summary of all these different sorts of origins of TSEs. So here we have people eating BSE contaminated meat. And some of those individuals passed the disease on to other people when they gave blood or corneas or whatever before they were recognized to have the disease. We think the disease went from cattle to other cows, to a variety of animals who are fed cow meat, even to cats. Domestic cats fed cow meat as part of their cat food have been shown to get, poor kitty gets a TSE, right? Where did this all originate? Well, probably it started with spontaneous disease in sheep and perhaps elk. The minks might have picked it up from eating some sheep as well. And in people, it's probably a sporadic disease originally, and then passed on to other people by contaminated material. The last question is, which of the following illustrate the prion species barrier? Right, number 2 is the correct answer. Mice fed hamster PRP-SC do not develop disease unless they have the hamster transgene. That illustrates the species barrier, not any of the others. Now, I just want to leave you with the important fact that not all prions cause disease. There are prions in yeast and other fungi that don't cause disease, but they can switch conformations just like the pathogenic ones. And these are thought to be epigenetic switches that allow proteins to have different functions and allow, for example, a yeast, if it changes its environment and there are fewer nutrients, it can alter a protein's function just by conformational switches. And there have been a lot of these discovered in yeast. These are the gene names, and these are the prion state, so different names depending on prion or normal. And then there's the function normally and the consequences of the prion state. So the prion protein that we've talked about today normally has a function in neuronal growth, but of course when it's conformationally altered, it's causing disease. But all of these other prions in yeasts have another function of the protein. For example, this URE3 gene is normally, URE2 is normally a repressor of transcription of nitrogen catabolic genes. But when it switches to the prion state, again, it's a conformationally altered state, just like PRPSC, it now allows utilization of other sources of nitrogen. So these are good things. The proteins can switch conformation and allow them to have other properties. So it could very well be that having this conformational switch ability is actually good in evolutionary terms. And for some reason, the PRPC-SC switch that may have a good function that we're not aware of, but most of the time it is leading to pathogenesis.